This is USMLE QA. USMLE material presented in a question and answer format structured similar to the first aid for the USMLE Step 1 book.
Question: What is the TCA cycle also known as? Answer: The TCA cycle is also known as the Krebs cycle. Question: What is the first step of the TCA cycle? Answer: The first step of the TCA cycle is the conversion of pyruvate to acetyl-CoA. Question: What is produced during the first step of the TCA cycle? Answer: The first step of the TCA cycle produces 1 NADH and 1 CO2. Question: What does the TCA cycle produce in total? Answer: The TCA cycle produces 3 NADH, 1 FADH2, 2 CO2, and 1 GTP per acetyl-CoA, which equals to 10 AP per acetyl-CoA. Question: Where do TCA cycle reactions occur? Answer: TCA cycle reactions occur in the mitochondria. Question: What cofactors are required by the α-ketoglutarate dehydrogenase complex? Answer: The α-ketoglutarate dehydrogenase complex requires the same cofactors as the pyruvate dehydrogenase complex, which include vitamins B1, B2, B3, B5, and lipoic acid. Question: What is the starting substrate for making oxaloacetate in the TCA cycle? Answer: Citrate is the starting substrate for making oxaloacetate in the TCA cycle. Question: How many AP molecules are produced per acetyl-CoA in the TCA cycle? Answer: 10 AP molecules are produced per acetyl-CoA in the TCA cycle. Question: How does the TCA cycle generate AP? Answer: The TCA cycle generates AP through the process of oxidative phosphorylation, in which the energy from the electrons carried by NADH and FADH2 is used to pump protons across the inner mitochondrial membrane, creating a proton gradient. The energy from this gradient is then used to generate AP through the activity of the AP synthase enzyme. Question: What is the role of NADH and FADH2 in the TCA cycle? Answer: NADH and FADH2 are produced during the TCA cycle and are then used in the process of oxidative phosphorylation to generate AP. NADH carries electrons from the TCA cycle to the electron transport chain, where it donates them to the electron carriers and generates AP. FADH2 carries electrons from the TCA cycle to the electron transport chain, but generates less AP than NADH because it donates electrons at a later stage of the chain. Question: Why is the TCA cycle also called the Krebs cycle? Answer: The TCA cycle is also called the Krebs cycle because it was discovered and described by Hans Adolf Krebs, a German biochemist, in 1937. He received the Nobel Prize in Medicine in 1953 for this discovery. Question: How does TCA cycle get electrons to generate AP? Answer: The TCA cycle gets electrons to generate AP by breaking down acetyl-CoA, which is derived from glucose, through a series of enzymatic reactions. These reactions result in the production of NADH and FADH2, which are then used in the process of oxidative phosphorylation to generate AP. Question: How does the TCA cycle contribute to the production of CO2? Answer: The TCA cycle contributes to the production of CO2 through the process of decarboxylation, which occurs during the first step of the cycle. When pyruvate is converted to acetyl-CoA, decarboxylation releases CO2 as a waste product. Additionally, during the process of oxidative phosphorylation, the electrons carried by NADH and FADH2 are used to pump protons across the inner mitochondrial membrane. This process also consumes O2 and produces CO2 as a byproduct. Question: How does the TCA cycle connect with the process of respiration? Answer: The TCA cycle is a central component of cellular respiration, a metabolic process that converts the energy stored in glucose and other organic molecules into a form that can be used by cells. The TCA cycle converts acetyl-CoA, derived from glucose, into CO2 and generates electrons in the form of NADH and FADH2, which are then used to generate AP through the process of oxidative phosphorylation. Question: How does the TCA cycle connect with other metabolic pathways? Answer: The TCA cycle connects with other metabolic pathways such as the citric acid cycle and the electron transport chain. The citric acid cycle is a series of chemical reactions that converts acetyl-CoA into CO2 and also generates electrons in the form of NADH and FADH2, which are used to generate AP through the process of oxidative phosphorylation. The TCA TCA cycle also connects with the process of beta-oxidation, which breaks down fatty acids to produce acetyl-CoA. Question: how does the TCA cycle consume oxygen? Answer: the TCA cycle consumes oxygen during the process of oxidative phosphorylation, which is the process by which the energy from the electrons carried by NADH and FADH2 is used to pump protons across the inner mitochondrial membrane, creating a proton gradient. This process consumes O2 and produces CO2 as a byproduct.
Thank you for watching our Q&A format video. We hope this helped you learn, review, and retain knowledge needed for the USMLEs. For more information on this topic, check out the link in the description. Now, if you want to take your USMLE prep to the next level, be sure to check out all our courses, which are structured to follow along with the First Aid for the USMLE Step 1 book. You can find us at usmleqa.com. And if you're watching this on YouTube, be sure to like and subscribe.